I don't, is your background also in computer science or just in biology and psychology?
No, computer science first, then biology.
Okay, all the more so. Michael is the director of the Allen Discovery Center at Tufts University, which is also Dan Dennett's former institution. His lab works at the intersection of developmental biology, artificial life, bioengineering, synthetic morphology, and cognitive science. Seeking general principles of life as it can be, that's they, I'm reading their statement, they use a wide range of natural animal models and also create novel synthetic and chimeric life forms. Their goal is to develop generative conceptual frameworks that help detect, understand, predict, and communicate with truly diverse intelligences, including cells, tissues, organs, synthetic living constructs, robots, and software-based AIs. Their applications range from biomedical interventions to new ethical roadmaps. Welcome to Montreal, Mike, and it's all yours.
Thank you so much. And thank you for having me at this amazing gathering. It's a real privilege to be able to share some ideas with you all. If anybody's interested in the primary papers, the data sets, the software, everything is at this site here. Okay, so there's a few things that I would like to say today. I'm going to just kind of give you an overview right up front. I'm going to talk about this multi-scale competency architecture, which underlies most of biology, and in particular, some very interesting physiological software that is operating on this genetically defined hardware. We're going to talk about some concepts, including the cognitive light cone, or the size of the largest goal that a system can pursue. We'll talk a little bit about polycomputing, or this idea that the same biological processes are interpreted by different subsystems as very different computations. I'm going to go in some detail into an example of an unconventional basal intelligence, basically collectives of cells that expertly navigate anatomical space and solve all sorts of problems. This has many implications for biomedicine and bioengineering. Towards the end, I'll talk about the idea that the synthetic morphoengineering advances provide really an enormous option space for new bodies and new minds that do not have standard evolutionary backstories. So this really offers us an opportunity to think about evolution, biomedicine, robotics, and ethics in new ways. And we in my group focus on emergent cognition, not just emergent complexity. So the bottom line to all of this is that biology definitely does things very differently than today's AIs and robotics, but I don't think it's specifically because of brains. I think it's way older than that. And I don't think there's any particular reason that we cannot make use of these same properties in our artificial constructs. So this is an old engraving. This is Adam naming the animals in the Garden of Eden, and something like this underlies a lot of discussions in this field, even if not sort of consciously acknowledged. There are two things represented here: one that I think is deeply wrong, and one that is actually quite profound, and it's going to matter a lot to us. The thing that I think needs to change is this idea that all of these are natural kinds. That there are discrete kinds of animals that are very sort of unique, specific things, and this atom here is the most unique of them all. And we can talk about human intelligence as a specific demarcated thing. The thing that I think they got quite right is that in this story, it was on Adam to name the animals in the Garden of Eden. Now, this is interesting because according to these ancient traditions, naming something means that you've discovered its true inner nature. And I think, as you'll see by the end of the talk, I think it will be on us to name a whole wide range of novel beings that are going to be living with us, and to understand their nature and to be able to live with them in a kind of symbiosis. So the first thing we can tell from work of really classical scientists, well over 100 years ago, is that this idea of a human, a normal human, which features prominently in discussions of philosophy and AI and so on, is really at a certain point in a very smooth continuum. So both on an evolutionary level and at a developmental level, all of us evolved from single cells, and we developed from single cells. And so when you make claims about what humans do or do not do, we have to ask ourselves where in this continuum those capacities emerge, how do they scale, those kinds of things. Even more complex is the fact that we now know that at every level of organization, we can introduce novel, either biological or designed material, which means that there's also a continuum this way of technologically modified or biologically modified beings. So whatever it is that we're saying about humans in terms of their capacities, their moral responsibilities, and all of those kinds of things, we need to ask how far away with really quite small steps you can go along all of these different continua, and how do those things change as you do that. So the framework that I'm working on has as a goal this aspiration to help us recognize, create, and ethically relate to truly diverse intelligences, regardless of what they're made of or how they got here. And so, of course, this means familiar types of biologicals, apes and birds, and maybe an octopus or something like that, but also some very unusual creatures. This means colonial organisms and swarms, engineered new life forms, AIs, whether purely software or some sort of robotic, and potentially at some point even exobiological agents. And the criteria for this framework is that it cannot be just philosophy. It has to move experimental work forward. It has to, these kinds of perspectives that I'm going to talk about today have to, the acid test is, do they help us make new discoveries? And I'm going to argue that they do. They actually lead to novel research programs. Of course, I'm not the first person to try for something like this. So here's, Wiener and colleagues back in 1943 that was trying to understand what the scaling of relationships are like, all the way from passive matter up to the kind of complex metacognitive properties that we have. And the details of all of this are in this paper if you're interested. So one way to think about different kinds of systems, really diverse kinds of systems, is to adopt an engineering approach and simply ask, what are the tools in our toolkit to optimally interact with each kind of system. So at one end, you have simple machines where hardware modification is the only game in town. Then you have these sort of cybernetic kinds of things like homeostats, where you can actually change the set point and rely on the system to do some work to hold on to it. Then, of course, we have systems that have different kinds of learning and memory, and then all the way up here and whatever's beyond that. So one of the things that I think is true about these kind of continuum models is that, first of all, they take all cognitive claims to be protocols, communication and relationship protocols, as opposed to statements about the system itself. What you're saying is how you plan to interact with that system. That's I think what you're really saying when you say that a system is somewhere on this continuum. And then what's good about that is that it's an empirical claim. So you can try it, see how that works out, and somebody else who thinks the system should be at a different position gets to try their bag of tools and see what happens. So we use this approach to ask, for example, where do groups of cells in your body, where do they fit in? And the current assumption of, let's say, molecular biology is that they're here, maybe edging into this, but I actually think that's wrong, and we do experiments to try to sort that out. Now, in this audience, I don't need to introduce Alan Turing to anybody, but what's really interesting about what he was doing when he was interested in problem-solving machines and intelligence through reprogrammability is that he also wrote a paper. This is one of the earliest papers of modeling the processes of embryonic development on how order arises in the chemistry of early embryogenesis. Now, you might wonder, why would somebody who is interested in mind and intelligence and computation and things like that, why would he be interested in what's happening with chemicals during development? And I think, although he didn't write much about this, but I really think that he was onto a profound truth, which is that the origin of minds and the origin of bodies are fundamentally very symmetrical processes. So what I'd like to do in the next few minutes is to take you through a tour of some of unique features of the biological substrate. There are some very interesting things about biological materials and their capabilities that I think shed light on this process of origination of and scaling of minds and what it means for understanding and for AIs in general. So the first fact about biology is that it really makes this journey. So all of us were once a quiescent oocyte, an unfertilized oocyte, a little blob of chemicals. And slowly, very gradually, over a lengthy time period, it's self-assembled into one of these remarkable functional anatomies, or maybe it's something like this that's then going to make claims that it's not a machine and it's different from the what the laws of physics allow. But this is the journey that each of us undertook, and so we have to understand what's happening here. What exactly is going on when the mind arises from this kind of humble beginning? Now, another thing that we might think is that, okay, so we arise from a single cell. Clearly, the kind of processes of chemistry and physics are our origin, but at least we have a centralized, unified intelligence. We have a brain. We have a solid brain, not like ant colonies or swarms of bees, which some people, Ricard Soleil calls them liquid brains. But a lot of people think that's just a metaphor. But we actually have a nice, unified, centralized brain. And in fact, Descartes was really into the pineal gland, because there's only one of them in the brain, and it was tempting to think that our unified experience is in some way responsible, is related to this singular structure. But of course, if he had access to good microscopy, what he would see is that there actually isn't one of anything, because inside the pineal gland are all these cells, and inside of each of those cells is all of this stuff. So in an important sense, we are all collective intelligences. We are all made of parts. And the interesting story here is how those parts give rise to higher order cells. So this is the sort of thing. This is a typical building block. Now, this happens to be a free living organism called the lacrimaria. Single cell, no brain, no nervous system, no stem cells, just a single cell. Very competent in its local, tiny little sphere of influence, aka its cognitive light cone is quite small. The goals that it pursues are very small. Now, what's remarkable about the biological material is that it can individuate higher order cells from large collections of cells. So let's see how that works out. So here are embryonic cells of a human, let's say a human, for example, embryonic blastoderm. There's maybe, let's say 50,000 cells at this point. That's a flat sheet. We look at this and we say, okay, there's an embryo, and we know what it becomes. It becomes one human with a human mind. Now, what is it that allows us to look at 50,000 cells and say that's one of anything? What makes it one embryo? Well, what makes it one embryo is literally alignment, both physical alignment and functional alignment. All of these cells, they are connected. They are all going to align via planar polarity, and they have all adopted the same picture of where they are going to go in anatomical morphospace, meaning they are all going to work together to build exactly one structure. They are all aligned with respect to their anatomical goals. They are all going to do one thing. Now, what you can do is you can take a little needle, and I used to do this as a graduate student in duck embryos. You take a little needle and you can make scratches in this blastoderm. And what will happen then is that until for the next, let's say 4 to 6 hours, until these scratches heal, each one of these little islands does not feel the presence of the others. It self-organizes into its own embryo. It is aligned within itself, but not with these others. And when they finally heal up, what you end up with is multiple embryos in the same blastoderm. So this is how twins, triplets, and so on arise. So what's happening here is that each one of these embryos has to demarcate itself from the outside world. They have to ask, where do I end and the other self or the outside world begins? Because every cell is some other cell's neighbor. So establishing borders is critical. And this kind of splitting, this fragmentation, has all sorts of interesting relationships to things that we see in cognitive science in terms of dissociative identity disorders, split brain, and so on. There's something else here, which is that having seen this blastoderm, you cannot say how many individuals there are, because that number is not fixed by the genetics. It could be 0, 1, 2, 3, probably up to half a dozen. The number of cognitive selves that come from this individual, from this blastoderm, is not fixed in advance. They self-organize in real time. Okay, another important thing about the biological substrate is that it really does not overtrain on its evolutionary priors. It is very flexible and willing to do new things. So for example, here is a tadpole. There's the mouth, the nostrils, the brain, the tail. For millions of years, this creature has evolved to be successful with two eyes here connected to the brain. What you'll notice here is that we've prevented the primary eyes from forming, but we've induced an eye on its tail. I'll talk more about how we do those things. So it turns out that this animal can see perfectly well right out of the box. We made this machine. It's an automated training and testing tool that trains them in visual assays. These eyes do put out an optic nerve. That optic nerve does not connect to the brain. It connects to the spinal cord, to the gut, or sometimes nowhere. And despite that, these animals perform very well in visual assays. So you don't need new rounds of evolutionary adaptation. It self-assembles a functional form, and the behavior adapts to this new architecture of the sensor motor system. It also, the biological substrate also enables information to move across the media in really interesting ways. For example, here's a flatworm. You'll hear more about this in a bit, but one of the things about them is that they regenerate. So you can cut them into pieces, and every piece gives rise to a perfect little worm. So what happens is that if you train them, and they have a true centralized brain and most of the same neurotransmitters that we have, if you train them in a place conditioning task to recognize a particular area where they will look for food, you can then cut off their head, leave the tail. It does nothing for about 8 or 9 days until it grows back a new head, and then behavior resumes, and then you find that they remember the original information. So what's happening here is that presumably the information is stored elsewhere in the body, but then it's imprinted onto the new brain as the brain develops. So you can see here a crossover between the morphological memory that leads them to rebuild the correct structure, but also the behavioral memories that can be stored outside the brain and then re-imprinted onto the new central controller here. There's an important aspect to this, which is that that information is not merely stored in the sense of kept intact, but it's actually remapped. And so let's consider this kind of system. You've got a caterpillar. The caterpillar has a particular brain suitable for navigating a two-dimensional world and eating leaves. It has to turn into this. This is a soft-bodied kind of creature. This is a hard-bodied creature, flies in the three-dimensional world, drinks nectar. Completely different controller needed for that. And so what it does in the meantime is basically liquefy its brain. Lots of the cells are killed off. Most of the connections are broken. And yet what's been shown is that these moths or butterflies remember the original information. But what's interesting here is that the exact memories of the caterpillar, meaning crawl in a particular way for a particular stimulus to get leaves, are of no use to the butterfly at all. The butterfly does not crawl, it doesn't care for leaves. So what has to happen is that this information has to be remapped to make sense in a completely new body architecture in a new environment. And so one way to think about this is that biology does not emphasize fidelity of the information. What it emphasizes is saliency. In other words, if you think about memories, even our own memories, as a kind of message from our past self, the idea is that those messages, like any message, have to be interpreted. The compressed engrams of past experience have to be interpreted by our future selves in whatever way is most appropriate to the context. I think biology fundamentally assumes that the hardware is unreliable, both in a developmental context and evolutionarily. Not just the environment will change, but your body will change. You will mutate. Everything is going to change. And this is why these kinds of things like the tadpoles with the eyes on their tails and some other things I'm going to show you, this is why they work. Because out of the box, the system does not assume that it can take the information literally. It has to do a construction project that is going to use the past memories in whatever way it can, but reinterpret them as it has to. And so all of that is described here. So another important thing for these kind of the discussions of AI and so on has to do with embodiment. So it is often said that what you really need for true cognition is some kind of body that's going to act in space, and that you can learn from that. But actually biology uses all kinds of spaces that are different from the three-dimensional space that is so obvious to us. So we're reasonably good at recognizing intelligence in familiar three-dimensional space, but that's mostly because we're so used to it, and all of our sense organs point outwards, and we recognize medium-sized objects moving at medium speeds as typical intelligences. But biology operates in many other spaces. So they saw living things solve problems in the high dimensional space of gene expression, in the space of physiological states, and what we're going to talk about today mostly is anatomical morphospace. So there are these other spaces that are not visible to us except through experiments. They are not easy for us to sort of visualize and reason about, but perception action loops can absolutely happen in all of these other spaces. So just because something doesn't have a body in the conventional sense of some sort of robot that drives around the physical world does not mean that a system is not doing all of these kinds of things that are necessary for cognition in some other space. In fact, I would argue that what evolution has done, and I'm going to show you kind of this middle example here, is pivot some of the same navigational strategies across spaces. So first metabolic space, and then physiological space, and eventually genes came on the scene, and there was transcriptional space, and then multicellularity, and you had anatomical space, and eventually brains and muscles showed up, and now three-dimensional behavioral space, and even navigating linguistic space. So what we have then in biology is this multi-scale architecture, but it's not just a set of nested dolls structurally. It is the fact that each of these levels solves problems. They have their own competencies in different spaces, and they use some of the same tricks actually, but at different levels of sophistication. And so what I'd like to do now is to talk about one kind of deep dive into one case study, which is the collective intelligence of cells and how they solve problems in anatomical space. And so just to kind of back up again into the big picture, what we're looking at here is an unconventional intelligence that we are going to try to use some of the same tools of behavioral science, of neuroscience, and so on, to understand how we can use those tools to give us greater prediction and control over a system that is doing some very interesting problem solving. I'm going to show you some biomedicine that was discovered by attempts to communicate with this intelligence through its preferred interface, and I think this has real applications because in the sense it takes neuroscience well beyond neurons, and we can use this to try to push AI beyond really kind of neuromorphic architectures that seek to mimic the brain. So this is an example. So this isn't AI or Photoshop. This is a real insect. This is a fly. It has this kind of it's sort of running a very low resolution, stripped down two-dimensional morphogenetic program of some ants on its wings. The goal is to deter predators. So it sort of does this little dance where it waves the wings around and it looks like ants are scurrying around. And so that's pretty remarkable, but it's not as remarkable as the formation of the actual fly itself. Where did this complex structure come from? So this is a human torso. This is the cross-section through a human torso. You can see the amazing order here. All the structures are the right shape, the right size, relative to the right thing, and so on. But we start life as a collection of embryonic blastomeres. So where is this pattern actually encoded? How do the cells know to do this? And people are tempted to say DNA, but as we've known for a long time, and especially now that we can actually read genomes, the DNA doesn't directly specify any of this. The DNA specifies proteins and the tiny molecular level hardware that every cell gets to have. All of this, meaning knowing what to build, knowing when to stop, is a consequence of that hardware playing out physiological kinds of activities. And we need to know, as workers in regenerative medicine, we need to know how to induce cells to rebuild it if there's damage. And as engineers, we have a further question, which is what else could they build? How much plasticity is there here? Like what else could we get perfectly normal, genetically normal cells to build? And the end game of that effort I view as something I call the anatomical compiler. So the idea is that someday you will be able to sit down in front of this machine and draw that plant or animal or organ or biobot or whatever it is, draw it at the level of anatomy, not the molecular biology, but the anatomy. And then what the system would do is compile that description down into a set of stimuli that would have to be given to specific cells to get them to build whatever it is that you want them to build. Now, on a practical level, the reason this would be nice is that birth defects, traumatic injury, cancer, aging, degenerative disease, all of these things would go away if we knew how to communicate our goals to a set of cells and get them to build what we want them to build. And of course, keep in mind, this is the idea here. This is not a 3D printer. The idea is not to micromanage the construction of these things. It's a communications device. It's fundamentally to communicate your goals as the bioengineer to the collective that then has what I'm going to show you momentarily, which is a kind of homeostatic set point. Now, the thing is, we don't have yet anything remotely close to that. Molecular biology is now very good at this kind of information, which molecules and genes control which others. We're very far away from control of large scale form and function. And I think that's because currently most let's say molecular medicine has been stuck at this stage where everything is at the hardware level. So genomic editing, protein engineering, pathway rewiring, all the exciting advances have been at the level of the hardware. But what we've barely touched really is the most important and exciting thing about biology, which is the reprogrammability and the intelligence of the material that we're dealing with. Now, when I say intelligence, what do I mean? I like William James's definition, which is a nice substrate independent kind of definition, which says it's some degree of the ability to reach the same goal by different means. And he talks about the limitation of barrier of magnets not being able to go around a barrier because they're not smart enough to temporarily go against that gradient, all the way up to, you know, Romeo and Juliet separated by all kinds of barriers that they have complex cognitive skills to try to get around. So now we can ask this question. Okay, so on this continuum, given this amazing biological material that has problem solving at multiple scales, lots of plasticity, what kind of intelligence does it have? What problems can it actually deploy? Well, the first thing we notice is that while development is very reliable, so almost all eggs end up the correct anatomical shape, it is actually not hardwired, because what you can do with embryos of many species, including humans, is cut them into pieces, and you don't get half bodies. What you get are monozygotic, perfectly normal twins, triplets, and so on. And so that means that they can navigate this anatomical space from different starting positions to the same ensemble of goal states corresponding to the normal human target morphology. They can avoid various local maxima along the way. There are many examples that we could talk about if we had more time. But the point is, the process is not hardwired. In a few species, actually, it seems to be, but the vast majority of animals are not like that. It has this ability to get to where it's going, and that in some animals does not stop at the embryonic stage. This is an axolotl, and this amphibian regenerates its limbs, its eyes, its jaws, its heart, portions of the brain, spinal cord, and so on. And what's amazing is that if you cut the limb at any point along this axis, the cells rapidly spring into action. They build exactly what's needed, no more, no less, and then they stop. That's kind of the most amazing thing about all of this, is that they know when to stop. When do they stop? They stop when a correct salamander arm has been completed. That is the set point that they reach, and then activity ceases. Now, by the way, amphibians are not the only ones who do something like this. Other creatures can do it too. We are sort of not great at it. We regenerate our liver. Human children regenerate their fingertips, and deer regenerate huge amounts of bone, vasculature, innervation every year. So we can do a little bit of, but it's that ability to restore what you have to get back to that same location in anatomical space, despite unexpected perturbations, being dragged into a region of that space that you did not expect, is not the end of it. There's something even more interesting. So this is one of my favorite examples. So what you have here is the lumen of a kidney tubule in newts, and usually there's about 8 to 10 cells that get together and they form this little tube. So one trick you can do in these animals is to make the cells much larger by increasing the copy number of their genetic material. If you increase the genetic material, the cells get bigger. The first amazing thing is that you still get a normal newt out of it, even though they have multiple copies of the genome. But what you see is that fewer cells are now getting together to build this exact same structure. What you can even do is, and so the second amazing thing is that they scale the number of cells due to the novel cell size. What you can do is make a 6N polyploid newts, which have huge cells. And what these cells do, there's not even enough room for 2 cells here. So what they'll do is they'll wrap around themselves and still get you the same final structure using a different molecular mechanism. This is cell-to-cell communication and tubulogenesis. This is some kind of cytoskeletal bending. So in the service of an anatomical goal that they're trying to make, they can call up different tools in their toolkit, different molecular processes to get to the same region. So this is just imagine what it means if you're a newt coming into the world. You can't depend on how much genetic material you're going to have. You can't know what the size of your cells are going to be. You don't know what the number of your cells are going to be. You have to be able to finish that journey in morphospace in very novel circumstances. I think biology, and specifically evolution, assumes from the get-go that we cannot count on any of this. And so what it's done is really produce algorithms that are able to complete these journeys even when things change. Not just the environment changes, but actually the internal parts change. And that competency, that ability to fix problems and get to where you need to go, even if your starting material is different, produces what I think is a real intelligence ratchet, because it hides information from selection, and it forces evolution to do most of the work on the competency mechanism instead of the structural genome. And you end up with species like planaria that are immortal, cancer resistant, highly regenerative, and have an incredibly chaotic genome for reasons that we can discuss in the Q&A. And so the final example that I want to show you before I tell you how all this works is this example of navigation that has to do with changing from a tadpole to a frog. So normally, here's the tadpole. They have to rearrange their face to become a normal frog, and so all the organs have to move. And typically, it was thought that this is a hardwired process. Every organ just goes, in the right direction, the right amount, and it works. So we decided to test that, and we made these so-called Picasso tadpoles. So everything is in the wrong place. The eyes on the back of the head, the mouth is off to the side. Everything is scrambled, and these animals still become quite normal frogs, because the genetics does not specify a set of hardwired movements. What it actually specifies is an error minimization scheme. It specifies a system that can start off in the wrong position. All of these organs will move in novel, in fact, abnormal paths. Sometimes they go too far and they have to come back. And eventually it settles down into a correct frog face. So what we're thinking here is that, in addition to the standard story of cell biology, which is that there's a kind of this open loop emergence. So complexity and emergence comes from lots of local rules being executed, sort of like a cellular automaton. Actually, that's true, and that does happen, but that's not the only thing that happens. There are these feedback loops, which, if you are deviated from your target morphology, there are these feedback loops that try to reduce the error. So this is fundamentally a homeostatic circuit that tries to get back to where you need to be if you're deviated by mutation, by injury, by teratogens, and so on. And so this raises some obvious questions. For example, where is the set point stored? How does the system know what the set point is? Every homeostatic system should have a set point. Where is that set point stored? And of course, this is a pretty unusual thing for biology, because of course, we know all about set points for pH, for temperature, for the hunger, things like that. But here, what you have as a set point is actually a pretty complex, not a single number, it's not a scalar, it's a complex representation of the anatomy that has to serve as the target state for this process. So that ability to reduce error, to reach your target morphology, to navigate that anatomical space, is the intelligence that we're talking about here. And as I've shown, you can what it's able to do is to use different underlying mechanisms to get its goals met. So we now, a couple of decades ago, started the search for where is the memory for all of this. And we took our inspiration from neuroscience, where we have the kind of uncontroversial example of a set of cells that store a memory that guides future behavior, right? So the goal directedness. We know what this looks like in the brain. So here's the cellular hardware, the network of cells with ion channels that create voltage states that may or may not be propagated to their neighbors. And then the physiological signal processing that happens here is, according to the commitment of neuroscience, is the medium in which the memories, the goals, the preferences, the capabilities of that creature are encoded. Everything is in the electrophysiology of the system. And then the sort of neural decoding project is this idea that if we could read it, and so here's a zebrafish brain in a living fish, if we could read this information and decode it, we would know the cognitive content of this mind. We would know what the animal was thinking, what it remembers, and so on. Well, it turns out that this architecture is very powerful, and it was invented long before brains or even neurons came on the scene. This is ancient, about the time of bacterial biofilms is when evolution first discovered that these electrical circuits are really good for storing information, for making decisions, for integrating information across distance and also across time. Every cell in your body has these ion channels. Most cells have electrical synapses connecting them into a network. And what we started to do was basically the same thing, this idea of decoding these bioelectrical states to try to understand what does the what do the networks of the body think about. We know what the networks in your brain think about. They think about moving you through three-dimensional space. It turns out that long before that, what they were used for, these electrical networks were used for to move the configuration of your body through anatomical space. Okay, very similar kinds of tricks projected into a different space. So we developed the first tools to read and write this information in and out of tissues. So we use voltage sensitive fluorescent dyes. You can see that here in cells. The colors represent voltages. These are not models. These are real data. This is an early frog embryo as the cells are trying to figure out what shape are we making, how many eyes, who's left, who's right, that kind of thing. And then, of course, we do a lot of computational simulations, both biorealistic ones, where we try to understand where the voltage patterns come from, and then things like pattern completion and what happens when part of the pattern is actually gone. How does the collective reset it? I want to show you a couple of examples. Here's a time lapse of a frog embryo putting its face together, and what you will see here is one particular snapshot out of this voltage imaging, where what you can see long before the genes turn on to regionalize the face, you see here's the memory of what a correct face is supposed to look like. Here's where the eye is going to be. Here's the mouth. Here are the placodes. These cells already have a pretty good idea of where everything goes. And not only do the individual cells communicate via this pattern, but actually individual embryos. You can see here this wave passing between embryos. And here you can see the different cells kind of sorting out what they're going to do. These are the kind of this is analogous to brain imaging. This is where we're actually seeing the electrophysiology that that is responsible for the anatomical problem solving that you saw before. Now, how do we know that this is functionally instructive? Well, we developed tools to actually now read and to actually write modified electrical information back into the cells. Now, we do not use any kind of electric fields. There are no waves, no frequencies, no magnets, no applied electromagnetic radiation. What we do is basically take advantage of the interface that the cells are using to hack each other. Cells tell each other what to do via electrical signals through their gap junctions and through these ion channels. And that is the communication interface we exploit by putting in and taking out various channels, by optogenetics, which is using light to turn these ion conductances on and off. Okay, so what can you do if you start manipulating this bioelectrical information? Well, one thing you can do is you can activate very high level anatomical subroutines. So we take some RNA that encodes a particular potassium channel, we inject it into cells that are normally going to be this gut here. And what that ends up doing is saying to these cells, build an eye right here. And that is what they do. They build these eyes. The eyes have normal lens, retina, optic nerve, these kind of things. What we do not do is provide all the information about how to build an eye. We are not micromanaging this process. We do not directly control the gene expression. We are not talking to stem cells. We expect the material to know how to build an eye. And what we are providing is a stimulus that gets it to do that. Now, as it does that, it has many other competencies. For example, if we only target a few of the cells, so this is a cross section through one of those eyes, if we target only a few of those cells, what they do, so the blue cells are the ones that we targeted, all of this other stuff that's participating, that was, they induce their neighbors to help. Okay, so that's a secondary instruction that we do not have to teach them how to do this. And we say build an eye, they can tell there's not enough of them to build a proper eye, and they recruit their neighbors the way that other collective intelligences scale their numbers to appropriate tasks. This has obvious kind of regenerative medicine applications. So we've done this kind of thing for regeneration of limbs. So frogs, unlike salamanders, do not regenerate their limbs, but so that's what normally happens. But if you use voltage modulation to tell these cells that, hey, you should go down the leg building route instead of the scarring route, then you can trigger within 45 days, you've already got some toes, you've got a toenail, eventually a touch sensitive and motile leg. That's a pretty good anatomy. You can get 1.5 years of leg growth after just 24 hours of bioelectrical communication with those limb cells. After you do it, you don't need to touch them again. The system takes off and takes care of the rest. And I have to do a disclosure here because Dave Kaplan and I have a company that is going to try to push this into human bioengineering, but the idea and try to regenerate limbs in human patients eventually, hopefully. But the idea here is that asking these kinds of questions about what is the material capable of, what is the causal architecture, what are the subroutines that we can access, leads to all kinds of new discoveries. It leads to preclinical work, hopefully eventually real biomedicine. And the last example of that I want to show you is about really reprogramming that hardware. And so this is the planarian. So these are those flatworms that I told you about. And these guys have some amazing properties. They are resistant to injury because they're highly regenerative. Every piece will regrow into a perfect worm. They are, at least this asexual strain, is immortal. They do not seem to age. They are completely resistant to any kind of transgenesis. So no one has a transgenic mutant kind of planaria, unlike other species of which, of course, you can get many mutants. And that's because this species, more than any other that I'm aware of, is assuming that the DNA is unreliable and has remarkable algorithms to build the right structure, even if the hardware is modified. It has a very chaotic genome. And the one way you can modify it is by changing the representation of what these tissues think a correct planarian should look like. And I'm using all those words on purpose. So here's how this works. Here's a planarian. You cut off the head and the tail. By the way, this animal, here's what the normal molecular biology looks like. Head markers in the head, not in the tail. Very reliably, it gives a one-headed worm. Now we discover, now you can ask the question, how does this middle piece know how many heads it's supposed to have? It's not as simple as a positional information gradient, because these cells to the left of the cut line and the cells to the right of the cut line are in the same position along the animal, but they have radically different anatomical fates. This will make a tail, that will make a head. So how do they know? So it turns out there's an electrical circuit that stores a pattern that tells them how many heads to have. This is the pattern. So this is the voltage pattern: one head, one tail. What we can do is we can rewrite that voltage pattern. It's kind of a mess because the technology is still being worked out. But what you can do is you can say, no, actually 2 heads. And if you do that, and then you cut this animal, this is what you get. Again, not Photoshop, these are real animals. Now, what's important is that this voltage map is not a map of this two-headed creature. This voltage map is a map of this perfectly normal looking, one-headed, anatomically normal, molecular, biologically normal creature. What you can do is you can revise the stored set point, the memory of the collective intelligence of these cells, as to what it's going to do in the future if it gets injured. Until it gets injured, nothing happens. It sits here normal. Once you've injured it, then it consults the pattern, and then that is what it builds. So this is actually a really primitive counterfactual memory. I think it might be an early version of that amazing time travel that we have in our brains, where we can remember things and imagine things that are not true right now. This is a simple version of that in the collective intelligence of these cells. It is now we can actually see it now, right? We can actually see, how does it know how many heads to build? This is how there's a permanent a stable bioelectrical state that tells it what it's going to do. Now, the reason I keep calling it a memory is because if you then go ahead and recut these two-headed worms, you will find that they continuously regenerate two-headed with no further manipulation. Nothing has been done to their genome. There's no genomic editing. There's nothing wrong with their genetics. The hardware is completely a wild type, but the pattern, the memory of what they should be building has been permanently altered. And now they're going to continue to do this, and we can set them back. So you can see a little video. Here's what these, two brains in one body, lots of interesting, neuroscience to be done there as well. And so we're doing a lot of computational work trying to understand how the biophysical properties of the electric circuit map onto some of these amazing properties of memory, of pattern completion, and so on. Not just the number of heads, but actually you can cause them to grow heads for different species. So this triangular headed species can be made to grow flat heads like a P. felina, round heads like an S. mediterranea, whoops, the shape of their brains and the distribution of their stem cells become just like these other species, between 100 and 150 million years distance between these guys and them. And what's happening here is that in anatomical space, you can get that same hardware to visit different attractors in anatomical space where these other species normally hang out, but it's perfectly happy to go there if that's what the bioelectric pattern memory says. It is highly reprogrammable and it holds, as I showed you in the two-headed worms. Now let's just take a look at the limits of that plasticity. Not only can we push them to make shapes of other species, but we can get them to make shapes that planaria don't normally make at all. So you can ask them to, sort of larger distances in this latent morphospace. You can get these spiky things, cylindrical kind of combination forms. The cells are perfectly willing to build. Now, interestingly, we are not the only bioengineers that humans are not the only bioengineers that try to hack these competencies. Here's one of my favorite examples. So acorns make leaves that look like this, and you might think that that is what the oak genome knows how to do. It knows how to make these leaves. But in fact, there's another bioengineer, which is this wasp. And what they do is that they use chemical signals to prompt these cells to build something completely different. So these are galls. These structures are made of the plant cell. They're not made of the wasp cells. And the wasp is not genetically engineering them. What it's doing is hacking the normal morphogenetic competencies of these leaf cells to build something completely different. Now, if you hadn't seen this, would you ever have any idea that these things, which extremely reliably make flat green structures of a particular shape, could ever make this spiky red and orange kind of crazy thing? So the question is, what are the competencies of any given biological hardware, and what can it actually do? And so for the last few minutes, I want to talk about this idea that because of this architecture, you don't just get emergent complexity, you get emergent cognition. And so we're going to talk about cancer for a couple of minutes, and then we're going to talk about some synthetic life forms. So the one thing that happens here in both developmentally and evolutionarily is that little tiny homeostatic loops, for example, pH regulation, can scale up through policies that we can talk about into large systems that have very large goals, right? So this by being connected, these homeostats become connected in networks, and they're able to represent larger and larger goal states. So this is what's happening. While these single cell creatures have very tiny cognitive light cones, they're only trying to implement goals that are about the size of a single cell. The rest of it is just environment. Evolution and development gives them some great properties that allow them to now pursue very grandiose, large-scale goals like this, like stay in this region of anatomical morphospace. If you're deviated, come back to it. So these are very large. So no individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely does, because if you try to deviate it away, it will come back and then stop when it's done. But that ability to scale your cognitive light cone to connect into electrical networks that represent larger and larger goal states has a failure mode. That failure mode is cancer. So when the electrical connections are broken, what you see here is a reversion to unicellular ancient behavior. This is human glioblastoma. These cells have disconnected electrically from each other and from normal tissue. As far as they're concerned, the rest of the body is just external environment, and they've rolled back to their tiny little ancient goals of metabolic, physiological, and proliferative kinds of states. So that view of cancer as a kind of dissociative identity disorder of the collective intelligence of the body makes a very specific prediction. It says that cancer is not more selfish, it just has smaller selves. That what's happened here is that the boundary between self and world has shrunk. And so the idea here, why this is important, is that the size of the mind of the collective agent, what are the goals that it can pursue, are not fixed, and at least in biology, they're not fixed. They can grow and shrink even within the animal's lifetime, or the creature's lifetime. And so what you can see is using this bioelectrical imaging, you can see that, okay, here's where we injected the human oncogene. It's going to have a tumor. Even before the tumor appears, you can see where that's going to happen. And now we get to find out whether this crazy way of looking at the biology, really looking at cancer, not as a genetic disorder or a mutation and hardware that needs to be killed by toxic chemotherapy, but literally by a dissociative kind of condition. That leads to a prediction that actually, once you do this, once you inject these oncogenes, you don't have to kill the cells. What if we just forcibly reconnect them to the rest of the body? Because the first thing that transformed cells do is electrically decouple from their neighbors and become amoebas, basically. So when you do this, and so we've done this in the frog system, now we're moving into humans, human cells, and eventually human patients. What happens is that this is the same animal, even though the oncoprotein is blazingly expressed, it's all over the place. There is no tumor. And that's because it's not the genetics that drives, it's the physiology. It's once these cells are electrically coupled to their neighbors, it's the collective that works on building good organs, good skin, whatever muscle, whatever it's going to build, instead of this individualized behavior as the boundary between self and world shrinks. So that's the thing about biological hardware, is that it has this amazing multi-scale competency architecture where the parts can rebel, because that boundary between self and world is so dynamic. It gives us our plasticity, the ability to handle novel circumstances, problem solving in ways you've never seen before. But it also means that your parts are they run the risk of rebelling, which is typically not something we see in today's robotic architectures, for example. They're kind of have a much more flat. So what we see is that this architecture can set the size of goals, and now we can ask at the very end, we can ask, where do these goals come from? What sets the goals? So I'll show you this little creature. So here it is, running around, and I might ask you what this is. And you might guess that this is something I got off the bottom of a pond somewhere, some sort of primitive organism. If you were to sequence the genome here, what you would see is Homo sapiens, 100% Homo sapiens, no change. These are cells taken from an adult, in fact, elderly human patient. They are tracheal epithelial cells. In a new environment, they can reboot their multicellularity, and they can make something completely different that does not look like their adult form, nor does it look like any stage of human embryonic development. We call them anthrobots. They have all kinds of interesting capabilities. They are self-motile. They swim around with these little cilia. Really interesting, many interesting properties. I'll just show you one. One thing they know how to do is, if you grow some human neurons in a dish and put a big scratch through it, like a wound, like this, these bots will sit down. Here's a super bot cluster. There's a bunch of them sitting here. And what they'll do right here, if you lift them up 4 days later, you see that what they've done is taken the edges of the neural wound and healed them together and sewn them together. Now, who would have known that your tracheal cells, which sit quietly in your airway for decades, have the ability to self-assemble into a motile little proto-organism with a different set of behaviors, with a very different transcriptome, actually, and has these weird competencies. There are others. These are our xenobots. So these are made of frog skin cells, no neurons, just frog skin cells. And one of the many things they can do is take loose skin cells that you provide them with and collect them into little balls, both collectively and individually. They push them together into these little balls, which then mature and become the next generation of xenobots. And guess what they do? They run around and collect more and do the same thing. This is we call this kinematic replication. It's sort of a simplified version of what von Neumann wanted, which is a robot that makes copies of itself from stuff it finds in the environment. And so all of these competencies, these different things they do, we really need to understand where these things come from, because where they don't come from is eons of evolutionary selection. There's never been any xenobots. There's never been any anthrobots. There's never been direct selection for the kinds of things that they're doing. These are consequences of the material, of the competencies of the material, that are able, through all the different examples that I've showed you, are able to reconfigure themselves into functional beings in novel environments, and that degree of plasticity gives us phenotypes that we could not have predicted at the beginning. We do not have a science that would have looked at the genome for sure, or even at the cellular properties and told us that something like this was going to happen. But actually, the reality is that you don't have to go far to find unexpected problem solving. It does not take complex biology. So this is a preprint. The paper is in revision right now. Even something as simple as a classic sorting algorithm, we're talking bubble sort, selection sort, things like that, with if you look at it the right way, if you watch how it navigates sorting space, you can find some very interesting properties. And I don't have time. It looks like I'm running out of time here, but it has very interesting properties that are not directly in the algorithm. It has some problem solving capabilities that are not at all obvious from the algorithm, and apparently nobody has noticed it despite the decades of work with these algorithms. So my point,
Mike, don't worry about the time. The next session will be your panel. And so if you need more time to present the subject matter, it'll be taken up in the panel. Don't worry about it.
Okay, cool. Yeah, I'm almost done anyway. Just a couple of things. Yeah. So I mean, the lesson that I think we learn from this is a few things. First of all, in biology, you know, when we see these complex kinds of biological systems, you know, even cells are incredibly complex. When we see them doing new things we didn't expect, the temptation is to say, well, there's a mechanism for it somewhere. You know, we just haven't discovered all the mechanisms. Who knows what the selection forces were really? There's got to be a mechanism for it. But in extremely minimal systems, so there are people working in minimal active matter, so droplets and things like that. And then here, which is even more minimal, which is 6 lines of code, you know, in these sorting algorithms, fully deterministic, nowhere to hide, no new mechanisms. Even there, you see unexpected, not just complexity, not just unpredictable features, but unexpected problem solving in specific spaces. So my claim is that what we really need, and this is what the field of diverse intelligence aims at, is to really have a lot of humility about what the parts can do in terms of novel problem-solving competencies, and we should get better at noticing those. We should get better at understanding what space they're working in, you know, and things like even something as simple as a distributed sorting algorithm can do delayed gratification of the kind that James talked about. You know, they can temporarily unsort the string to have gains later when you start breaking numbers that refuse to move and things like that. You know, you confronted with challenges. You know, the typical sorting algorithm never asks, if I say swap 2 numbers, it never asks, did you really get swapped or did the hardware fail? And as long as you don't let the hardware fail, you don't see any of these things. When you see it is precisely when you're trying this with unreliable hardware, and then you see how it handles these problems. And I think biology is all about figuring out new ways to solve problems, because the hardware is known to be unreliable at every scale from the beginning. So because of this, because the biology is so fundamentally committed to plasticity and to not taking too seriously the meaning of past information, whether those be memories or genomic information and so on, it is also incredibly interoperable. So almost any combination of living material, engineered material like nanomaterials and electrodes and things like that, and software is going to be some kind of agent. And many of these already exist. I mean, there are already people that are, for all intents and purposes, cyborgs. There are hybrids. There are many different kinds of ways to combine living material and non-living material, which is critical, because a couple of things. This points out a couple of things. First of all, that old categories of, you know, this is a real organism and this is a mere machine, that doesn't work anymore. I mean, it never really worked, but for sure it's not going to work in the future, because any combination of these things is viable. And whatever theory you have about what organisms can do and what machines can do has to be able to handle all of the in-between cases. It is not just about evolution. It is not just about creatures that are on the tree of life somewhere with us, such that we can sort of know how to treat them because we know what they're made of and how they got here. Those kind of categories are not going to work anymore. So this is the kind of thing you have to have in mind when we talk about the capabilities of living things and of novel constructs. It is basically it is no longer feasible to just think about the set of evolved natural creatures that we have on Earth versus these kind of architectures that we make. Now, it is true that as I've pointed out, there are many aspects of biology that are very different from the way we engineer today. So the way that we make our hardware extremely reliable, the way we isolate the layers from each other, the flatness of our architectures, the commitment to fidelity of information storage as opposed to constant reinterpretation. You know, the fact that we see confabulation, the creative interpretation as a bug instead of a feature. Biology uses it extensively as a feature. All of those things are quite different, but there is no reason, I think, that we couldn't learn the lessons of what the biology is trying to teach us and make similar systems, or in fact, hybrid systems. And that is what I think the next few decades are going to be all about. It is also critical that the old standby for ethical decisions, you know, it used to be that confronted with something, you can sort of knock on it. And if you hear a clanging metallic sound, you can conclude that, okay, this came from a factory, it's going to be boring and predictable. And most of all, I'm ethically justified in, you know, refactoring it into a vacuum cleaner. I could do whatever I want with it. But if you had kind of like a soft, you know, sort of woolly kind of response, then you say, this is a natural creature and I need to be nice to it, and so on. So those kinds of that reliance on what you look like and where you came from is really not going to be feasible at all, even in the absence of actual aliens. But just all of the things that we are going to be living with, all of these novel bodies and novel minds, which means that we need better categories, better ethical categories of how we are going to relate to creatures that are nowhere on the tree of life with us. So this is the summary of what I tried to say today, that I think that intelligence in the sense of problem solving, I didn't say anything about consciousness, but in the sense of third person observable problem solving in different spaces, which are not obvious for us to recognize, is ubiquitous, especially in biology. It is everywhere. And we should be very suspicious of claims that something is not intelligent, because I think you know, the identification of intelligence is a difficult task. And I think it says as much about the observer making that claim as it does about the system itself, whatever limitations they think there are. We really, I think it's really essential, not only for biomedical progress, because learning to communicate with the cellular collective intelligence has enabled some really important advances. But also the ethical flourishing of beings is going to require us to learn to identify intelligence in places where we're just not familiar with it. I think it's really important to have some humility about the fact that we do not know what something can do, what it wants, what kind of goals it has, what are the scope of its goals, just because we know the algorithm, we know the material it's made of, or even because we made it ourselves. I think there's a lot of this is emergent in a very strong sense, and not just emergent complexity, but emergent problem solving competencies and emergent goals, which is really, you know, what makes this an existential level problem for humanity going forward. There's a research agenda here, which many people in the diverse intelligence community are following, and I kind of encourage anybody interested in AI and these bigger questions to familiarize themselves with the work of the DI community. We are looking for principled frameworks that avoid the typical teleophobia, this idea that if something is, you know, made of well-recognized materials, then it doesn't have real goals. It has some kind of derived goals from humans or something. But this idea that we can't just assume that these unconventional minds don't exist, nor are we able to daydream that every rock has a spirit hidden in it. We really need to continue this empirical testing of hypotheses about goal seeking and navigational competencies for diverse systems. So this is a very practical research agenda. This is not philosophy. And I think the future really, the kinds of conceptual approaches that are going to enable all of this to happen are a continuum of mind. So not binary categories of natural kinds, which we now know developmentally, evolutionarily, and via bioengineering are not crisp categories, and observer relative models that understand that any kind of cognitive claim that we make about systems is basically just it's an engineering protocol claim. It's a statement about how you think you can best interact with that system, and multiple observers will see different things, and then we can find out who's got the best approach. And we are working on the use of AI tools as actually a kind of universal translator to some of these diverse intelligences to help us communicate with them. So okay, I'm done. If anybody's interested, here are some papers that go into all of this in detail. And I want to thank all the people. So these are all the students and the postdocs who did some of the work that I showed you today. Lots of technical support staff, some wonderful collaborators, funders, all sorts of people have supported us. Again, disclosures. There are 3 companies that have supported some of this work, and most of all, I always thank the animal model systems, because they're the ones who do all the heavy lifting. So thank you. I'll take questions.
That was fabulous, Mike. I'm lucky enough to be in the privileged position of the moderator who can ask the first question. And I think you've shown compellingly a kind of continuity among functions in living, and also you spread it all the way to not yet living organisms or parts of. But the real question then, or at least the fundamental question that makes it an issue of cognition and not weasel words like intelligence, problem solving, those are things that it's easy to believe that even the primitive notion of a machine, which is a metal gadget, you can easily see how those things can become mechanisms that solve problems, that do it, and that we're ready to call intelligent. But the real borderline, and this is where I'm afraid of your answer, because everything so far has been intellectually and scientifically solid. But now I'm gonna, and you said you're not doing philosophy. Absolutely, you're not doing philosophy, you're doing biological science. But now we go into a question which is also not philosophical, but it's only philosophers that ever really give it serious thought. That little creature you showed with the long nose that could do all these, etc. What's that called?
It's a lacrimarian.
Okay. According to Mike Levin, is that lacrimarian sentient or not?
What is the definition that you're using? What
it can feel. It can feel. It feels like something to be that creature. It doesn't feel like anything to be my iPhone.
Well, yeah, I mean, so okay, so yeah, so absolutely, I think the lacrimaria can feel. If that's yeah, I mean, all of those words need definitions too, and here we get into, like, we can have a really tricky discussion. But I think I understand what you're asking, and I think there's no question the lacrimaria fits that description.
Do you think the brain is if we had to localize feeling in an organism, I assume you don't think that every bit of an organism feels. Would you agree that it's neural tissue that is the organ of this capacity and not just the somatic entity, or do you?
Well, a couple of things. So first of all, I don't believe any of these questions have binary answers. So I don't think we can ask, does it or doesn't it? I think it's a continuum, and I think we have to say how much and what kind. But I also, I mean, look, if we try to say, let's try to make a list of all the reasons why we believe that brain tissue is the seat of whatever term you want. I think we can make a pretty good case that lots of other tissues in the body meet that criterion. And I do actually think that there are other things that are absolutely not brains, including in our bodies, that have very sort of primitive basal cognitive experiences in other spaces. Yeah, I do believe that.
And who's feeling that? Am I feeling that if I have some in my stomach?
I mean, right. So when you say you, so right now your left hemisphere and my left hemisphere are having a lovely chat, but there are other components in your body that do not have the ability to verbalize their primitive basic feelings. But for the same reason that our brains can support this, yeah, I absolutely think that there are other organs in your body that have it. They just can't talk about it.
And I don't. So I'm like a split brain. I don't know about these other feeling creatures in my body.
Certainly, and you don't know about mine either, right? So we give each other the benefit of the doubt, but you cannot directly feel my inner perspective, and neither can you feel the direct. Neither can your left hemisphere, which is who I'm talking to right now, can feel the others in the body.
Absolutely, I've used up my prerogative now. That's no more from me. It's open to you, and please take advantage of it. He's absolutely remarkable. Do you have? Are you good at multitasking? Can you look up your? I could read them out, but can you look up the spectators' comments? Because
is this on the Q&A window?
yeah, it's on the Q&A window, and I can ask
it.
Julia to give hers a lot in words as the first one. Julia, do you want to voice your comment?
Sure. Thanks for the talk. Great. I was wondering, do you see a connection between the evolution of electrical circuits for storing information within organisms, which was something you touched on in one of your slides, and one of the proposed triggers for the development of human language, which is displacement, because they both seem to me to be related to trying to time travel.
Yeah, I mean, so okay, I do not have any evidence that these other systems that we work on, organs, tissues, whatever, that they have anything like human language. So we've not seen any evidence of that, and so I'm not claiming that. However, I think if I understand, when you say displacement, you mean temporally, right? Is that what you mean?
Yeah, like the ability to call up and refer to something that's not physically present.
Yep. I think we've seen primitive versions of that. I don't think we've seen anything like what what humans are able to do with it. But we have, yes, the need. I mean, even unicellular organisms can predict the future. They have a little bit of an anticipation horizon. That's been shown, and being able to answer what am I going to do if a specific thing happens is something that we see these systems doing in anatomical space. So if you were to take that to its logical conclusion, would you develop some kind of language that works in anatomical space? Maybe. I don't think it's impossible, but we don't have evidence for that right now.
Okay, can you see the comments there? Just in case, I'm going to do another call. If you have a question or a comment, either panelist or attendee, raise the hand or type it into the comments. There are two anonymous ones that you might want to handle. You've already done, Julia.
Okay, let's see. Striking similarities between your theories and Henry Lebroy. I don't know. I have to think. It's hard to say which of these things are necessity and which are coincidence. But I really do think that feedback loop, that ratchet that starts with the idea that your substrate is unreliable, and therefore the surviving the adaptive strategy is to be able to have to solve problems on the fly. I think that is what fundamentally kickstarts this spiral that we're so advanced at. Right from the beginning, the need to deal with novelty and perturbation, and to keep track of a certain path in physiological space and anatomical space, and eventually to keep a coherent linguistic story, which a lot of my understanding is a lot of LLMs have a problem with, is to keep a narrative going, for long periods of time. I trace that all the way back to the beginning of life. So I tend to think this is really a foundational force, if you want to call it that.
Henri Laborie didn't do anything faintly like what you're doing. I think I know who asked that question, and one can admire Henri Laborie, but not compare it with this. This is genuine biology, and it delivers what it's saying. I don't know. It's odd. I mean, everybody's gobsmacked by your talk, but it seems to have silenced them. I
could read a couple of the others. I see some of the other Q&As.
Go ahead.
Right. Let's see. Are the findings reconcilable with reductionism? Well, so I think reductionism is a frame that one takes when dealing with these systems, and I definitely think it's a very limiting frame, because the thing that I think reductionism is really good at is looking backwards. So if if I give you a system or an experimental result, then you can always tell a molecular level story about what just happened. So explaining backwards, you can always say, well, look, I can see this, this molecule bound to that molecule, no surprise. But I think what science and life in particular are all about is going forwards. The question is, does that framing help you do the next interesting thing? Does it help you get the next new capability, new discovery? And in some cases, the reductionist approach works. In the vast majority of cases that we're interested in, I absolutely do not think it works. And I think that there are really fundamental control knobs and fundamental phenomena that are, if you do try to reduce them to their components, you miss everything that's interesting about what you're trying to do. So I think it is a frame that is useful in some cases, but certainly not universal. Let's see, have I tried doing this with externally imposed magnetic fields? So unfortunately, magnetic fields are not a great way to manipulate electrical potentials in resting. Like, you can trigger neuronal firing and so on, but they're not good for setting up these kinds of things. So unfortunately, magnets don't work. I should also say that, bioelectricity isn't magical in the sense that it's not the only thing that can underlie this kind of cognitive scaling like, it is one great example, which is why I talk about it, of how individual cell competencies become large scale anatomical competencies. But there are other kinds of cognitive glue that function at the molecular level to make cells. And no doubt somewhere else in the universe, there are other mechanisms that do it. So, bioelectricity is important for us to has a good example, but I don't think it's the only way to do it. Let's see. Okay, could I say more about AI as a tool for shedding light on some of these topics? Well, a couple of things. So in our lab, we kind of have this, hopefully, this loop going where we use AI to try to help us understand the patterns that we're seeing, but then we also would like to take some of the things that we're learning and make more resilient, really truly bio-inspired AIs that I think would have very different properties from what has been made so far. And in particular, using it as a kind of minimally biased, I don't want to say unbiased, but like minimally biased way to detect intelligence. We need tools. We need appliances and tools and those kinds of things to recognize these sorts of beings around us. We are not good at it. And you need some impedance match between what you're trying to find and what you have. And so I think physics only sees machinery and not mind because it uses voltmeters and rulers and low agency tools. And so minds are good at seeing other minds, but we have a lot of very limiting firmware that we've sort of inherited in our evolutionary history. And I would like to see if AI can help us notice these kinds of properties. And so we're using tools to try to analyze other systems, whether they be purely simulations or actual real biological data, to generate hypotheses, to say, look, if you hypothesize this problem space, then you could hypothesize this is the goal. And look, now you gain extra explanatory power, and now here's new control knobs that you can use. And so I think we need tools to help us. Let's see, how did you learn which voltage to trigger to make a morphology develop? Okay, yeah, the spiky shape, right. So there are two kinds of experiments that I showed you. Most of them came from observing natural bioelectrical states, their anatomical outcomes, and basically trying to work out the mapping. So we call this cracking the bioelectric code. So most of them are like that. The spiky thing was not like that. The spiky thing, I have no idea. Like we guessed that, and it's basically us trying to probe the edges of this latent space. We have no idea. In cracking this code, we only know a few words in that code, really. I think this is just the beginning of a massive project. We could not have predicted that the spiky thing was going to form. The other things were predicted, the spiky form not. Let's see. Stefan, should I keep going? Or
yes, please. We'll cut it off at 3 o'clock and give you a little after 3. Give you a rest because it's gonna go on afterwards. I've been copying them down too, so we can import them into the panel.
Cool. Okay. Yep. I can do a few more. Let's see. Anything common to the dynamics of the commutative signal and cell networks and the dynamics of functional brain signals? Yeah. Great question. Yes. In fact, you know, Gorol Soel, who's a collaborator of ours at UCSD, has been recording signals from bacterial biofilms. And there are papers, you know, on brain-like activity in biofilms where what's happening is that the individual bacteria are using electrical signals to coordinate feeding across the whole, so that everybody in the colony gets to eat. So again, this kind of like cognitive glue thing. They actually use potassium spikes as a timing signal. So same thing with us. We are currently using analyses of these bioelectrical signals that are very similar to what people do in analyzing brain function. So calculations of integrated information and all of these other things. So, you know, I'm not prepared yet to say which are very similar and which are, you know, which are usefully similar. That is still in progress. But I think that, but we think there's enough there that we're putting a lot of effort into it. Let's see. We show that the flatworm's anatomy is controlled by the electrical signal across his body, and you can alter the anatomy during regrowth. How does the signal arise in the first place? Yeah, right. So that's a great question. We can watch the planarian is a funny case because planaria come from other planaria, but if you want to go to the origin, we can talk about the frog embryo patterns. So the frog embryo patterns we can watch from the unfertilized oocyte. Here comes the sperm, it gets fertilized, and we watch the pattern become more and more complex. The question of where does it come from is a tricky, it is a bit of a philosophical question. I mean, it's definitely not directly encoded in the genome because the genome doesn't say anything about patterns, the electrical or otherwise. The genome encodes ion channel properties. Now, in a certain sense, the genome certainly does contribute to the outcome because by constraining the specific electrical hardware, you get to constrain the space of possible dynamics. So in fact, it's more than that. You end up making hardware that reliably, by default, when the power comes on and metabolism cranks up, by default, it settles into a particular pattern. But what we see is that those circuits are also reprogrammable because you can change the pattern in all kinds of interesting ways, and then it can store it, and then it can change it in other ways, and so on. So where does it come from? I mean, look, we imagine a Galton board, right? So it's a board. It's got a bunch of nails banged into it, and you take a bucket of marbles and you sort of dump it in the marbles, go boom, 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 and then they land at the bottom. Every time you do this, they make a beautiful bell curve shape. So you could ask, where does the shape? Where is that shape encoded? You're not going to find it in the wood. You're not going to find it in the nails. Where is the shape encoded? So I think there are a couple of components here. I think asking where something is encoded sometimes is the right answer. For example, in asking for any given planarian, where is the current target morphology encoded, we know where. It's in the distribution of the voltage gradients. But also there is a sense in which it comes from wherever the laws of mathematics come from, because by establishing specific types of ion channels, you immediately get to use a bunch of what physicists might call free lunches, which is the fact that if you have a voltage-gated ion channel, what you have is a voltage-gated current conductance. You have a transistor. You have a couple of transistors, you can make a logic gate. When you have a logic gate, you immediately get all of the things that come with it, all of the laws of computation, the fact that NAND is special, like all of these things. You didn't have to evolve any of them. They all come for free, so to speak. And so part of these patterns come because evolution has shaped the hardware, and part of these patterns come because that hardware exploits the free gifts of the laws of computation, the laws of physics, the laws of geometry, and many other things. It's a really tricky question. But the implications of it are interesting. It's that, you know, what we're doing, and I think what these xenobots and anthrobots that I showed you, these synthetic constructs, what I think they really are exploration vehicles for exploring the latent space or the Platonic space, which evolution is so good at searching. I think what evolution is doing is searching for hardware that dips into this space of possibilities and exploits these different free affordances that are there. And I think what we're doing is putting a little periscope into that space and searching the area around that one point that a standard embryo gives you and tries to see what's there. Let's see. Would this framework
say the name out loud? Say the name out loud so I can keep track for what's left for you after.
Sure. The next one says anonymous, anonymous attendee. It says, would this
What's
framework?
the one before? That won't help.
Oh, sorry. Yeah.
What's the
Andrew
one just
Holiday.
before it?
Andrew Holiday. The one before that was Andrew Holiday.
Okay, thanks.
Right. Okay. So the next one's anonymous is, would this framework include human societies? So, but potentially, but I think it's really important to know that these kind of questions, I mean, people say, okay, then, our ecosystems, all of Earth, Gaia, the universe, the galaxy, are they also cognitive? The important thing there is that these are empirical questions. We cannot just have philosophical feelings about it. And by the way, I have nothing against philosophy. A lot of what I do, I think, is actually partly philosophy, but I like philosophy that's really grounded and that has practical implications. And so I think before we say anything about these other larger systems, we have to do experiments. Is it possible that some of these social systems, financial systems, I mean, so I have a collaborator that's actually testing this idea that the financial markets can be, trained and that and things like that. We have to do experiments, and the larger the system, the harder it is to do some of these experiments, but we can't say yes or no until somebody's actually done the experiments. I think that's definitely an empirical question. Let's see. Okay, next is also anonymous. It says, in cognitive neuroscience, the atoms of the current crop of normative models are perceptrons and learnable synapses. In your field, what would be the atoms of normative models? Well, two things. So at one level, the level that I talked about today, it would be the gap junction, which are basically it's basically an ancient electrical synapse that connects two cells directly. And I can tell you a whole story about why these gap junctions are an amazing kind of cognitive glue. But at a lower level, what I didn't even talk about at all is that you actually don't need cells to see some of these properties. Just signaling pathways. So imagine, I don't know, 5 to 10 molecules or genes that turn each other on and off in a network. That alone can give rise to 6 different kinds of learning, including Pavlovian conditioning. So it's really not tied to bioelectricity per se. It's tied to some basic properties of components that I think arise very readily at least in our world. They arise very readily and very early in what people think about the tree of life. I think I think this stuff predates life actually, as people define it. Okay, let's see. Okay, the last one. Yep. All right, okay, one more. That was perhaps more informative than I could process. Sorry. The story of cancer cells that suffer from a kind of communication disorder and develop a mini self as a result made me think about what role communication in all those different spaces has. Yeah, if I make other entities to communicate, might they unify? Yeah, I think communication is key. I mean, we've been studying different properties of the communication policies. So there are different policies that do or do not facilitate the formation of these kind of larger scale collectives out of pieces. It's not guaranteed. So, simply having a bunch of pieces connected to each other doesn't do it. We need to understand more about these policies. But yeah, I think actually communication is critical.
Do you want to knock it off and take a rest, or do you want to?
Let's take a rest and I'll and we'll come back. What is it, in a half an hour?
Okay, do me a favor before we turn this off. You should also copy into a text file the ones that are left from the next question, so that you have them to look at.
Okay, I will
just copy paste them into a text file.
Yep. Okay. Let's see. Is there any way? Is there an easy way to save them, or I gotta go?
Yeah, just mouse it into a. No, there is. Never mind. I have it, but then it'll force me to read it. But that's fine if you can.
I can do it, but it'll take me, some number of minutes to do it. There's a bunch more.
Trouble with that is that I want to save this video, and for that we have to turn the whole thing off.
Got it. Okay, I'm on it. I'm here. Let me do it. Yeah, boy, there's some great questions. I'm just gonna.
Good, no. You go ahead. I was talking to, I don't want to cut it off until you're done.
All right, I'm copying and pasting as fast as I can here. Oh boy.
We could scroll down the whole thing and then get it all in one piece instead of
Can
one.
you,
by one.
oh, I see, you're right. You can actually, oh geez, I didn't realize you could get them all. Thank you. Yep. Okay, 1 second. Okay. Okay, got him.
Okay, thank you. Cool.
All right.